Hello everyone, my name is Justin Howard. Today I'm presenting to you on discerning pigmentary proteins in chromatophores. I chose this subject for a couple reasons. One, I've already done a little bit of work over the last couple years on cephalopods and just their dramatic ability to change colors and their appearance on demand. And I've always found that fascinating. And also a particular study that I keep coming across and have read a few different times over the years. New stuff comes to light every time I read this study, especially after I've taken different classes, and analytical biochemistry was no different. This time around, I'm able to understand a little bit more about their liquid chromatography and how they separated these proteins within a chromatophore and really trying to discern how they work. And so what I want to review with you today, briefly, I want to go over just cephalopods in general, their taxonomy, phylogeny, where they fit at on the evolutionary timescale. A little bit about their nervous system organization, how that's comparable to mammals and other similar complex systems that are seen in vertebrates versus invertebrates. And then when we get down here to structural highlights of the organ, the proteins is when I'll really be talking about the study that I'm highlighting today. Hopefully I won't get lost, my thoughts won't get lost with this broad range of topics here. So just to orient us what we're talking about today, Phylum Mollusca is a class of invertebrates that contains this subclass here, this class Cephalopoda. And what we're talking about specifically today is the squid, octopus, and cuttlefish and the chromatophores that they use to change colors and appearance. These other things are on here for just reference at this point. Here we have a phylogenetic tree. That's kind of displaying how far removed these invertebrates are that I just mentioned compared to humans, for example, which have also developed a complex nervous system and with that behaviors and abilities. And so one of the reasons they think that these cephalopods continued to develop and organize such a complex nervous system was in response to predation from bony fish that were arriving on the scene about the same time that they were starting to emerge somewhere around 400 million years ago. And at least that's what most studies can conclude, was the time frame wise. And if you compare that to humans, which have only been on the timeline about 300,000 years, give or take, there's some interesting observations that come up in how they continue to develop these behaviors. And it provides for a good study model for convergent evolution, just based on their behaviors and things like that. Even though sometimes they're hard to untangle what's really going on. Are they making this color because they're mad, or are they making this color because they're trying to attract a potential mate? And but there are studies that are able to disentangle that in a lot of good ways. Still lots of work to be done, though, in that department. So just an overview here. In comparison, the human brain has groups of neurons that cluster together in what we would term as lobes. For example, our occipital lobe here receives signals from our retina, turns that into an image, flips it around so we can perceive it better. And those neurons in that lobe, where they're clustered there densely together, have that specific job. And here is an example of an octopus brain. And it has different lobes as well that are dedicated to different jobs. But what makes them a little bit different, if you look at this cross-section or anatomical section of an arm here in an octopus, it has this central nervous cord going down each arm. Let's look at the next slide here. And so what this equates to is multiple spinal cords with dense clusters of neurons able to perform specific jobs. Many times over, they have multiple spinal cords, basically, as opposed to our one, which is what we consider that with the brain, our central nervous system for humans. And what it gives them is basically a network-like structure for their nervous system. It's able to make decisions further downstream apart from that main processor, which we would say is the brain, because they have a lot more sophisticated neurons and clusters of neurons within these cords. Whereas ours is like a two-lane highway. We have information going to and away from this central point, the brain, to help us make decisions in response to external stimuli. And so because of that, they're able to do things like this. And actually, in this figure here, this video, this octopus is supposed to be dreaming. So I would count this as an autonomic response, one that's done involuntarily for the octopus through its nervous system. But they can also control it voluntarily as well. And so the molecules that give them ability to actually change color can be summed up as two different ones, basically. Pigments, which are contained in these nanostructures here that look like little spheres, and other proteins that can include like S-crystalline, omega-crystalline, reflectin, and R-opsin. These don't provide color, but they enhance optical properties of these tethered pigments when they are gathered together in this rope-like structure. And this rope-like tethering structure, that will be made up of some of these proteins here. These proteins are particularly special and found in not just the lenses of cephalopod eyes, but mammals in general, basically anything with a clear lens, because they will allow light to pass through a clear membrane, but still have properties that refract light, so that we can make sense of distance and things like that, depth perception. And as far as the organs that utilize those proteins we just mentioned, cephalopods aren't special, especially when it comes to these two, which I don't want to focus on too much. These iridophore and leucophores are found throughout nature. That's what gives peacock feather its iridescent properties and the way it reflects light, for example. But cephalopods go one step further with this organ called a chromatophore that can be color selective. And it is organized with a bunch of different tissue. So there'll be a sac in this chromatophore that contains the pigment granules we looked at earlier, and some of them will be tethered, some of them won't be. There's radial muscles here that will expand and contract this disc, like the pupil in your eye, sort of. And when it does that, when it expands and gets real flat, these granules become real thinly spread. And so based on how far apart they are and how thick they're stacked, gives them different abilities to reflect light and either absorb it or reflect something different. And right here in the video, you can see there's mainly red, brown, and yellow chromatophores in this squid section that they've gathered from skin here. And they're just expanding and contracting. This is sort of like another example of an octopus just on relaxed mode, where it's not really, it may even be sleeping as well. They kind of flicker like that. It's called a flickering pattern. And so finally getting to the last part of this here, these pigment selective organs, chromatophores, are usually layered in skin above or superior to these iridophores and leucophores that will be found underneath. And so what they end up being, one, I don't have it cited, but what one researcher wrote in his thesis is that these pigments basically are filters for the light coming from the iridophores and leucophores beneath. And so they are able to manipulate that light even further than what the iridophores have in order to change their appearance. Here's a good example of it in live motion. You can see the iridescent patchy spots. These are the lower layer of skin with iridophores and leucophores. And above them are the chromatophores. But take note that these chromatophores are in a circular pattern. There's red, brown, and yellow, basically, 3 colors of chromatophores. And they're all circling a patch of iridescent tissue underneath. And so that kind of lends to the idea that these chromatophores are manipulating light from the lens below, or from the iridophores and leucophores below. And what Williams et al. And his discovery team wanted to discern really was how exactly are these pigments changing this light that's coming from these iridophores, both structurally, and how much pigments are in play with that ability. And so what they did was take different sections of cephalopod skin from different cephalopods, all of the same species, and group them by color, one of these 3 colors here, red, yellow, or brown. And then they took all of those chromatophores and broke them apart protein by protein to understand what was going on with each one. And so when they did this right here, this first time, not only was the granules and the pigment sac included, but also this membrane on the outside called sheath cells. There would be some muscle tissue in there as well as nerve. And what they initially found through their tandem mass spectrometry with liquid chromatography was 469 total proteins, and they were able to identify about 412 of them as saying, yes, this is definitely a cephalopod or octopus squid genes, because they took those proteins they discovered and matched them up with the database, this UniProt non-redundant protein database, and said, yes, they're definitely cephalopods. And what they learned was that here they're grouped by biological function, basically. And so some of the proteins were for metabolism of the cell. Some were going towards making crystalline. They might be isoforms. Some of them were not characterized. Some are going towards making cytoskeleton, and so on and so forth. But these two right here, reflectin and crystalline, are the ones we're interested in. And so the results in terms of abundance was crystalline and reflectin were the first and second most abundant proteins within these chromatophores. And they had to go through and separate a bunch of different stuff that came out of these chromatophores. It was more than just 400 or so proteins identified, but these are the ones that were identified specific to cephalopods. Things like ribosomal leftovers from bacteria that may have been in the sample, they ruled those out with a long list in their supplemental information that was like 28 pages long of a table of things they found. And so trying to discern where this reflectin was coming from, they went back and just did the granules themselves. They washed all the pigment out of it and broke them down protein by protein and only found crystalline within the granules. So the big question is, where is the reflectin coming from? So what they ultimately discovered was that it was found consistently localized in the sheath cells outside of this sac here. And so when they were matching up the whole chromatophore and discerning what was inside of it, they were finding crystalline and reflectin. They did this 3 times just to make sure they were getting the right results also. And so how did they solve this issue of where the reflectin was actually residing versus the crystalline inside? Was with confocal microscopy, which we're going to look at next. I think it's a beautiful method for discerning some different biological structures. So if you've ever been sciencing like this guy with just a regular microscope in the lab, like in the classroom or something, you know that you can barely adjust this fine adjustment knob here and throw something out of focus. And so this is different focal planes within your microscope. But with confocal microscopy, those planes are all taken at the same time basically and put back together into a 3D structure if you need to. It can be coupled with fluoroscopy and certain cell markers, but you get these beautiful slides here with no blurred sections of it. It's all on the same focal length or focal plane. And there's one below it and one above it, and they, like I said, they stack these images up to get perfect non-blurry images. And here's a cross-section of chromatophore with the pigment sac inside. And then out here is this sheath cell that they are thinking will contain the reflectin. And it does. We're going to show how they proved that here in a second. But it completely envelops this chromatophore sac. And so no wonder they were getting both samples of both proteins when they were crushing up the whole thing and discerning what proteins were inside of it. And so another example where they fluoresced certain immunochemistry markers and then put this confocal microscope on it. You can see here our pigment sac and the radial muscles that will be flattening the sac or making it thick again by relaxing. Here they are relaxed, and the reflectin is bunched up adjacent to the sac. It's going to be on top of it and below it. And so they're able to make this image through confocal microscopy and make them all these planes in focus here. Here it is contracted, and this reflectin will be traveling out within this area here and then relaxed again. They've just shown it with different fluoroscopy markers here. It's back up and gathered around the cell when it's relaxed. And so what's going on is that this reflectin is contained within these sheath cells. Here's the radial muscles marked again. And as they expand, the sheath cell expands with it, and reflecting comes back, and then it's able to do its job, reflecting light through this pigment sac later. And so in conclusion, they were able to use liquid chromatography and mass spec to figure out what was exactly contained within the chromatophore and to systematically roll out all the proteins that could be manipulating light. And then once they discovered them, they were able to fluoresce them and mark them and actually view them in real time with some scanning electron images here that I've showed to you to understand where that reflectome really is within the cell. And made a pretty good discovery and sort of put the proverbial nail in the coffin on how these chromatophores were also working with iridescent light and not just changing color through absorption or reflection by themselves. I hope you all have enjoyed this presentation. It's not too long and easily followed. I wish everybody the best of luck in the rest of the semester and the rest of their time at WKU. Have a great afternoon.